Hello everyone, my name is Josie Rossbach and my presentation is on what is the relation between gut microbiota and COVID-19. Now you may be wondering, what is COVID-19? Well, COVID-19 is an illness that is highly infectious and unpredictable to how each individual will react, and it mainly attacks an individual's respiratory system. Some may have no reaction, others may have severe respiratory symptoms and long-lasting complications after this. So those with pre-existing conditions tend to have worse reactions to the virus, and many of those people that have been infected have died. Others recover quickly and live a normal and happy life afterwards. This variability is a factor in the fear of the disease, along with the idea that we know very little about it to begin with. Now, how does the gut microbiota relate to the respiratory system? The gut is almost connected to the lungs through something called the gut-lung axis. The gut-lung axis is a connectivity between the gut microbiota and the microbiota of the lung through the circulation of blood. For example, hyperinflammation caused from the gut microbiota can result in damage to the lungs, and lungs that have been infected from a virus can spread the infection to other organs through circulation. And as said in a journal by Dysbiosis and Abhishek Mohanty, the gut-lung axis is bidirectional, meaning the endotoxins, microbial metabolites can impact the lung through blood, and when inflammation happens, it can translate to the gut microbiome as well. An endotoxin is a toxin that is inside a bacterial cell and is released when the cell disintegrates, basically a lipopolysaccharide that regulates inflammation, located in the cell wall. Before we get too far into the understanding of the gut-lung axis, let's understand what the gut microbiome is. The gut microbiota is located in the GI tract. It hosts many different bacteria that play a very important role in the digestion and immunity functions. This variety of bacteria have lots of unique jobs in digestion and inflammatory responses. For a reference of amount, in a journal written by Bursar Akçez and Belma Aslim, the gastrointestinal tract hosts over 10 to the 14th power cells made up of 500 to 1,000 bacterial species. All of these microorganisms are referred to as the gut microbiota. Well, you might be wondering, what happens to the gut microbiota in reaction to COVID? Studies show that the virus notably decreases the amount of beneficial bacteria in the gut, and there's also an inverse relationship of this decrease with the severity of the virus per patient. Damage to the gut microbiota can result in slowed down digestion and the inability to digest certain nutrients. If the imbalance of the gut microbial community becomes severe, this can result in something called gut dysbiosis. This imbalance can cause inflammation and immune dysfunction. If one already has some sort of gut dysbiosis before COVID-19, their immunity will be low and they will have more severe effects of the virus. And patients that are recovering from COVID-19 have been found to have developed gut dysbiosis. Basically, the idea of gut dysbiosis is that it can lead to worse infection and it can be caused by a viral infection. As said in the article about the gut-lung axis and dysbiosis in COVID-19, dysbiosis in gut microbiota results in gut permeability, leading to the secondary infection and multiple organ failure. Conversely, disruption of the gut barrier integrity due to dysbiosis may lead to translocation of COVID-19 from the lung into the intestinal lumen via circulatory and lymphatic system. Side note, SARS-CoV-2 is just a scientific term for COVID-19. COVID-19 has been proven to be more fatal for the elderly and those with pre-existing conditions. As said here, elderly, immune-compromised patients, and patients with other comorbidities like type 2 diabetes, cardiovascular disorders fare poorly in combating COVID-19. Along with this, it was studied that the composition of intestinal microbiota is a major determinant in health and disease impact, immunity, digestion, and other functions. Individuals that have gut dysbiosis due to aging or other conditions are already at a disadvantage due to their lack of balance in their gut microbiome. Not only is this dysbiosis relevant to COVID, it has been linked to asthma, arthritis, obesity, type 2 diabetes, which correlates to insulin resistance. Overall, staying in balance is vital for health and the interaction between the gut microbiota and the immune system, and is crucial in determining the severity of COVID in an individual. Interestingly enough, another source of harm to the gut microbiome are antibiotics. Antibiotic treatment in patients with COVID-19 was associated with a more heterogeneous microbiome configuration and accompanied by a further shift of the gut microbiome away from a healthy microbiome. Overall, the gut microbiome of all patients with COVID-19 remained stable but were markedly disparate from that of a healthy control, both during the disease course and after clearance of COVID. This means that the dysbiosis caused by COVID-19 was shown not to be extremely detrimental, but the gut microbiome was in far worse condition than that of a normal GI tract. After a clinical trial, at the last follow-up, the gut microbiome of the 10 patients that were studied remained substantially different from that of a healthy control, despite clearance of COVID infection. Now that there is an understanding of what the gut microbiome is and how COVID is affected by its state and how COVID itself affects it, how does the gut microbiome affect the spread of COVID in our bodies? COVID uses the angiotensin converting enzyme 2, known as ACE2 receptor, to enter the host, and this receptor is highly expressed in both the respiratory and GI tracts. This idea relates back to the gut-lung axis and how the two microbiomes are correlated and affect each other. During coronavirus infection process, spike proteins of the virus recognizes ACE2 receptors to invade the host cell via type 2 transmembrane serine protease. Here's a visual to help the understanding of how the virus enters the host through the ACE2 receptor. You can see the SARS-CoV-2 spike protein binding to ACE2, and once the ACE2 receptor travels across the membrane, the virus has entered the host and begins to replicate. Since the ACE2 receptors are densely populated in the esophagus, stomach, duodenum, rectum, and along with the receptors in the respiratory tract, there are an immense amount of places for the virus to attack. There are many things an individual can do to help the gut microbiome integrity. To boost gut microbiota health, it is suggested to eat more foods that have dietary fibers, prebiotics like inulin and polydextrose, and probiotics like yogurt and kombucha. These foods will improve gastrointestinal health by creating more diversity in the gastrointestinal microbiome and help strengthen the intestinal lining. As previously said, the gut microbiome consists of many different bacteria and microorganisms that help us digest the food we eat and in turn use it. In the gut, healthy individuals are dominated by 4 phyla. They are called actiniobacteria, firmicutes, proteobacteria, and bacteroidetes. Further than that, bacteroidetes and firmicutes are predominant in the gut, while bacteroidetes, firmicutes, and proteobacteria are preponderant in the lung. We acquire these different bacteria through what we eat, along with environmental factors. As said here, different diets affect what's in the gut microbiome. Animal fat and protein-based diets versus vegetables-based diets are very different. High-fat diets that tend to be associated with protein-based diets have been associated with circadian rhythm disruption. Circadian rhythms is the internal 24-hour cycle of the body that regulates the sleep-wake cycle. Along with diet, environmental factors can modulate the composition of the gut microbiome. It has also been found that the consumption of the protein extracts of whey and pea increase gut commensal bacteria and decrease pathogenic bacteria. Dietary fibers are also a good source of microbiota-accessible carbohydrates, which helps intestinal health. It has also been found that the positive effects of fiber have been beneficial towards the lung microbiota, reinforcing the relations between the gut and lung microbiomes through the gut-lung axis. 2 very important beneficial things that we can eat that will help the gut microbial community are prebiotics and probiotics. Prebiotics are compounds in food that promote the growth of beneficial microorganisms. Prebiotics such as inulin, polydextrose, and maize fiber have been shown to improve the immunity, gut diversity, and digestion in humans and especially in elderly people. Good foods that have inulin are asparagus, garlic, leeks, and artichokes. Foods that have polydextrose are breakfast cereals and candies and desserts as a sugar replacement. Probiotics are live microorganisms that are edible that help improve and restore the gut microflora. Probiotics have shown good results in improving inflammatory conditions as well as regulating innate immunity using toll-like receptors and the corresponding signaling pathways. Some examples of good probiotics are yogurt, kombucha, and kimchi. And through the gut-lung axis, it has been found that many probiotics have been shown to improve or alleviate lung disease conditions. In conclusion, the gut microbiota plays a critical role in the digestive system. Having a variety of microorganisms within the gut helps the processing and digestion of all the foods consumed by an individual. The better the digestion of these foods, the more efficiently one can use the nutrients from the food, convert it to use as energy, or use it for anti-inflammatory purposes. The health of the gut microbiota has been found to be linked to the health of the lung microbiota as well through the gut-lung axis. With the development of COVID-19, having a good gut and lung health is essential in having moderate, non-lasting effects to the virus. The elderly and those with pre-existing gut dysbiosis have had more severe symptoms to the virus and sometimes are not able to recover. While evaluating the lung health previous to contracting COVID-19, it is important to note how the virus affects the gut microbiome afterwards. It has been shown that the virus significantly lowers the variety of the microorganisms that inhabit the gut microbiome, making the gastrointestinal tract weaker and increasing the susceptibility to other infections.